Yeah. Hi. I think it's something is happening. Let's see. Okay. Yeah. So it's recording now. Let's go to my question. Sorry about that. Yeah. So the question, any of you answered that answer to question two is C. Okay. So pollen grain and egg cells. Yeah. So pollen grains are haploid and egg cells are haploid. Yes. So next is the question is number three. Okay. Is it visible? I don't know. Hello? It's not visible, right? Okay, okay, okay. Yes, yes, yes. Yeah, just coming. Just give me 1 second of time. So I should be present it again. Yeah, sorry guys for the delay. I appreciate your patience. Okay. So let's see. So number three, I hope it's visible now, right? Yes. Okay. So the yeah. Yeah, thank you. So the question number three is that the dash generation is dominant in the bryophyte life cycles and that dash generation is dominant in seedless vascular plants. So what is that? Oh, which is so you know the alternation of generations, right? Yes. Mm. Yes. Yeah. So earlier, if you see the evolutionary, previously the gametophytic body was the dominant, okay, and and in the beginning of the development of the plant, if we say there were actually two individual bodies like gametophytic and sporophytic that used to develop, right? And then later on, it got much more congested. We can say that now the same plant body can actually harbor both kind of both the haploid and diploid. So now the one which you see is the major plant body would be the sporophytic, which thus the gametophytic will be the spores or the egg cells, which it will be bearing. Okay, unlike previous bryophytes, which had dominant body was the gametophytic. Yeah. So let's see. What is the question? Is a match the plant developmental process and the primary axis in which they occur. So activity of SAM and RAM, developmental process and the primary axis in which they occur. Okay, so are you guys clear about this axis, adaxial, abaxial? Is it? Yeah. Option B. Hmm. No, no, no. What I was asking, ki, okay, fine, that we can see whether it's option B or any other thing. The main thing is the concept of it, no? So I'm asking ki if you're clear about the adaxial, abaxial, the central, peripheral, these things or not. The axis, how it is done. Hmm. Okay. Shall I explain that? Shall I explain adaxial, abaxial, proximal, distal, this axis? Okay. Okay, let's go. Yeah. So if we think about the first one is the abaxial and addaxial. So suppose this is your plant axis. Okay, slides are visible, right? Drawings are visible. Okay. Now this is your axis. So this is your axis orientation. Now if I tell you that there is a leaf here, okay, and so if this leaf is supposed in a 45 degree angle to your main axis, then the surf so you know that leaf will be having two surfaces, like the upper one and the lower one. We'll know which one is what. So if if the surface is facing towards your vertical axis, that is the adaxial. Okay, so the upper one you can say is the adaxial surface, and the lower one, which is away from your vertical axis, is the abaxial. Okay, this is a term is basically used for the leaf, basically the adaxial and the abaxial side. Okay, another was that the I think the central and peripheral. So if you remember the shoot apical meristem, so if you see it in the from the top view, so basically the central is this one and the peripheral is this one. Okay, so this is your central and these are your peripherals. Okay, now proximal distal. Is this clear till here? Okay, so let's see for the proximal distal. It's very easy way to understand proximal distal is, so if someone is a human, then if you think of your arm, the 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 closer part of your arm to the body is the proximal, and the far away from it is the distal part. Okay. Now if we see the same thing in terms of a leaf, so if this is your leaf, then this region is going, if this is something, suppose this is your axis of you some branches in which you have this leaf, so this part should be the proximal and this should be the distal. So proximal is nearby, distal is the far away from it. Okay, yeah, I think one more was there. Let's see what is that. Yeah, apical basal. Yeah, apical basal is very easy, right? Apical, you know, just uh, something like in the apex is the apical, and something is the base is the basal. Okay, so let's see now. So what is the question is, first one is the activity of sam and ram. So what do you think, it should be adaxial, abaxial, central, peripheral, apical, basal? What is the case, proximal, distal? Apical, basal, sam and ram. Okay, apical, basal. Just wait. Let's see some more options. Activity of cambium. Cambium in secondary growth. So secondary growth is something peripheral. Okay, tell me what could be the option. Okay, fine. So development of upper and lower leaf surfaces. Just now I mentioned it. Abaxial, adductional, right? Yeah. So and last one is the branching. Branching, you can think of it proximal, distal, I guess. So if we think 3 as 1, 4 as 4, 2 as 2, that's what you mentioned, activity of cambium, central, peripheral. So 1 is going to be your 3. Yeah, so yeah, option B. Fine. Okay, so have you gone through this short root gene is transcribed in dash, but the protein is transported to this. So this is a very informational question. Truly speaking, I have not gone through it. If you have gone through it, let me know. I will tell you the bit of background of it instead. You have done the class. You have attended the classes, right? First one, okay. Short root is transcribed in vascular tissue, but the protein is transported to the endodermis. Okay, everyone is agreeing with the answer. Fine. Okay. So so I will tell you something else. If you already know the answer, it's okay. I will try to tell you the concept about it. So whenever you know that some kind of gene is being named, so how is it being named? So what earlier people used to do, if I say that, if you think of a screen in the sense, if they are putting or I mean treating the plants with some kind of mutagenesis, then they're able to get a different kind of mutants in the sense which has a altered phenotypic which has an altered phenotype from the wild type, the one which they're choosing. Okay, so those plants they're calling are mutants, so they have something mutated in them, and then they are choosing them based on some kind of phenotype. So here, if we think of the short root gene, so correct me if I'm wrong. So is that so? Generally, what happens that how do they name the genes is that whatever they see in the phenotype of the mutant. So suppose if I think of the short root may have a very short root length when they in the mutated gene. So they saw that okay, this gene is getting a specific gene, suppose X, is getting mutated, and that's giving rise to a phenotype, suppose short root. So they named it short root. Later on, this found that this is the later on after mapping QTL and all the other things, SNPs. So they found that they might have found that this is the gene responsible for that, or this is a segment of the chromosome is responsible, and they might have named it short root. So now let tell me, is it the case that short root mutants are having short roots? As I've already attended the class. Yeah. Why no answer? Okay, fine. Anyway, whatever it is, so someone is saying that it's getting transcribed in vascular tissue and being transported to the endodermis. So this is basically what I know about this gene is it's for the patterning of the ground of the radial tissues. So they help to form the so patterning in the sense there are different kinds of cells in a root tissue, right? So there will be the ground tissues, there will be the vascular tissue, so maybe the patterning of that it helps in. So if you remember, if you've heard of it, another gene if I talk about is the uh, apetala. Have you heard of it, anyone? Did I lose the connection or what? Okay. Yeah, so A petala is a mutant which does not produce petals, is it? Yeah. Anyway. So the A petala, A gammas, A petala. Have you read about the ABC flower model? Yeah. So there are certain mutants. So that's what I was just trying to tell you. Ki if there will be certain mutants which, if they lose a gene, then according to the phenotype, that specific is termed. Okay. So suppose if you remember any of it, that the ABC class of gene. So C class agamas is a C class gene. If you lose it, that so C class genes are actually helps in the formation of in the determination in the sense that when flowers are formed, no, from outside you can see the petals, then sepals, then androecium, then gynoecium. So when it reaches to gynoecium, the stem cells has to stop the division. Otherwise, what will happen if they do not stop? That can lead to the formation of another flower. So that's exactly what happens in the C class mutant, that they keep on producing the flowers. Okay, and they're also having some defect in the female reproductive development. So that's why they named as agamous. Okay. Anyway, next question is, okay, just mark it. I'll check the answer. The ability of a single plant cell to give rise to the entire organism is called, or yeah, I knew everyone will answer this question. Okay. Totipotency, I think, is the entire organism as mentioned. Pluripotency still can give rise to a different kind of cell, but may have certain limitations compared to the totipotency. So, which of the following is the chemical mutagen? Okay, which of the following is a chemical mutagen? Yeah, EMS. Okay. So, which, with reference to the TOS17 based transposon mutagenesis, which of the following statement is false? I have to explain two things here, transposon and, okay, one more thing. Yeah, any guesses? Yeah, yeah, option D is the answer. Okay, so what do you mean by retrotransposon? Yeah, then what is retro? Yeah, so what happens that it's retrotransposon is also a jumping gene. The only difference is that while retrotransposon, the DNA actually gets transcribed to RNA, and then again it gets I mean reverse transcript into DNA, and then it gets inserted into the target genome. So probably it was not getting through the RNA pathway, but here one RNA pathway is involved. Okay. Yeah, so this kind of transposon mutagenesis and EMS mutagenesis, let me talk about a bit of conceptual about it. So what happens is that EMS, you know, that causes some modification in the basis, right? So what happens that plants, not plants exactly, I mean the seeds are treated with EMS. So it's like thousands of seeds. It could be 5,000, 10,000 of seeds that gets treated with EMS, and then they sow all the seeds and they check for some phenotype which is altered. Phenotype in the sense the overall morphology, if we say, of a plant, and if you're comparing wild type to any of the I mean it's mutated with the wild type, and you search for any kind of defect, in the sense it could be from your desired what is it desired I mean whatever you are desiring to search for a phenotype. It could be in the leaf, it could be in the flower, it could be in the root, anything. So you see what you might be interested in a particular phenotype, or you can see as a general, you can see that this phenotype looks very interesting, so I want to further study about it. So this, even for the transposon, is the same thing. The transposons screens are also done, these are not targeted like CRISPR. These are all random mutations, so this can go and bind I mean it can go and target any sequence of the genome. You also know about I mean the gamma radiations. They are also the unbiased, I mean the biased, not biased, sorry, the non-biased, means they can just go randomly into the genome and cause some kind of mutation. So then when they get these mutants, so suppose you got a specific mutant, it looks something different from the wild type, okay, then you collect that or choose that, then you cross it with the parent, or the parent, or suppose the homozygous parent, you have some for that gene, and then you generate a F1 progeny. You know that if something is I mean, if something is is head. So in F1 you know that you will get a heterozygous condition, right? So what happens that they're not F1, they call it M1 anyway. So M1 is the mutant one generation. So in that M1 they will let them to self fertilize, and they will get a variety or a very big number of F2 generation, and then they will again search for that phenotype which they are interested, or in the first place they selected for. And then they do further mapping in the sense you can just think about the whole genome sequencing, and then see with those specific plants whether they are harboring any nucleotide polymorphism or not. If all of them showing the phenotype are having or harboring the same SNP or the single nucleotide polymorphism, then they further, with the help of different markers, they map to a specific gene, and then they get to know, okay, this gene might be responsible for this phenotype. Okay, so this doesn't end here, they further mutate that gene artificially again, and then see that whether they are able to reproduce that phenotype or not. Okay, so this is how different. So if you do thousands, so there are actually dedicated institutes, I would say, like if you heard of Teeling, ABRC. So they have this collection of not ABRC, we think about it about Teeling, they have the collection of huge point mutations in their stocks. So if you are interested in, suppose, in a specific gene, suppose X, and you want to see whether there is any mutant available in the Teeling harboring this mutation, you can search their database, and if you're lucky you can get one of them. So these are all, so what I'm trying to say that these are all just random mutations by which they can create one mutation at a time, maybe they can select for, but when they're doing multiple things together, they can have a collection of huge collection of genes which are mutated and have an associated phenotype with that. So can you, as I told about it, can you name something in which you can actually nowadays is very common as like, can you target a specific gene as such? Yes. I mean, my question, I'll repeat my question. Can you think, imagine of anything which can actually target a specific gene instead of this random mutagenesis, which we were discussing right now, is the chemical mutagen or transposon mutagenesis? Can you think of some technique which can specifically target a gene, a specific gene? Site directed mutagenesis. Anything? Yeah, those are fine. Yes. Anything else other than that, which is very common nowadays? You might have heard a lot. CRISPR, right? You're aware of CRISPR, right? Yes. Yes. Yeah, if you're interested, we can talk about that also. So, anything you know about CRISPR, like how does it work? Mm. Yeah. Then? Hmm. Yes, sir. Yes. 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 Yeah. So, is, you want me to talk about that also? Because as we're coming through that, I have a slide. Yes, yes. Yeah. Let me just. If I have the slide, I can talk about it. I don't think I have it. Unfortunately, I don't have the slide, but I can talk about it a little bit with the help of. Okay. Okay, so I don't have the actual one, but I'll just make use of this slide. So, yeah, so as I think Sir and some of you suggested that it's the guide RNA and the Cas9 protein, right? So, yeah, or you can say the enzyme because it's an endonuclease enzyme. So, what happens that if I talk about little bit about the I mean history of it. So CRISPR is basically we talk it as a tool, but it actually a system for the bacterial cell. So what they do is that it's a specific region in their genome that harbors sequences of the viral genomes. Hmm. So I'll just draw it. So it's Yeah. So in that genome, there are certain sequences of there are viral sequences as well. That is known as CRISPR. So something regularly interspaced sequence. I forgot the full form of it. So what is that? That whenever a bacteria gets infected by a virus, so their a system like this Cas, there is something other protein, other Cas proteins. What they do is that they cleave a part of viral DNA, and then they incorporate into their genome. Okay, and they do it in such a wonderful manner, I would say, that they keep the viral sequence and they put a spacer. Okay, then again another viral virus, when they gets infected, they cut that part again and then they put it again along with a spacer. So viral sequence spacer, viral sequence spacer, that's how they form a cassette, and that cassette is known as CRISPR for them. So what happens that this ones can form whenever they get transcribed, they can form this guide RNA, and then along with the Cas9 protein, when the same virus gets infects the E. coli, this guide RNA can go and bind to the complementary site of it, and then the Cas9, the endonuclease enzyme, can cleave the DNA sequence of the virus, or you can say the viral genome. So that's how they use it for their own defense mechanism. And if you are already aware of the CRISPR system very well, they actually require some more sites like as PAM sites, so that helps in the recognition of some of the genome sequence. Okay, and interestingly, what Charpentier and those group, the Nobel Prize winners, they have found later on that our human genome also has a specific PAM sites. Okay, that's have a specific sequence. So what they thought that if we can actually design the bacterial system to, I mean, the same way, if we can design some guide RNA along with the Cas9 expression system, if we can provide that sequence, so that sequence will be specific to the DNA sequence. Okay, so instead of and if that sequence has a PAM sequence, that will be even more for binding to that site, and then once they gets binded to that site, then Cas9 can actually cleave that part and lead to the, you can say, it can be a knockdown, it could be a knockout of the gene. Knockdown in the sense that you are actually reducing the expression of that gene, and knockout is completely, I mean, the protein is not formed at all because of the many deletions and all. Okay, so that's how the idea was. So this way, they're able to target a specific site of the DNA, or of a specific gene, we can say. Is it? But nowadays, what people are doing, that it's not the only way that you can. It's not that the nucleus is going and cleaving that part. They're also doing something else, like they can enhance that specific region by instead of cas9 they can use some other enzymes or other proteins which can actually help in the enhancement of the transcription of that region, or else they can use some kind of protein which will help in the suppression, or the reducing of the transcription of that specific gene. And if you're also aware about, as I'm talking about this slide, so if you're also aware about the epigenetic system, so what happens that that different gene has a different mark, like as methylation mark, acetylation mark. So methylation mark is a repression mark, acetylation is an activation mark, right? So but if we can somehow can provide some enzyme which can lead to demethylation or deacetylation of that specific region, that can also lead to suppression or activation of a gene, right? So there are nowadays is a different multiple ways. So yeah, but the overall thing is true that it is the gene specific genesis. And one more thing is, it's not, of course it's not the first one. Before that also there was something like talin, that were also gene specific. But the thing is, there were that system was a bit different because they were using basically proteins. So protein, we know that also can bind to DNA. So they were using specific proteins which can go and bind to the DNA, and then led to this activation or suppression, something like that. Okay. Yeah, is this fine? Yes, any question on this? Okay. Yeah. So next question, let's see. Yeah. So a molecular marker with which one is able to differentiate between a homozygous and a heterozygous condition is called a, yeah, codominant. And why you are saying it's codominant marker? Okay. Any other answer? Yeah, that's fine in one way. Okay. So how do we actually differentiate between a homozygous and a heterozygous? So fine, let's see, whatever it is, codominant, dominant, we'll just have an idea of how it works. Okay, I don't have that. Okay, but see, uh, if we talk about just let's see, have a general idea about how something, if you want to know how how a system is heterozygous or homozygous, how will you differentiate between that? So we know that homozygous is something which will have the same two copies of the same allele. Yeah. And another will be, one could be dominant, one could be recessive, right? That is in the heterozygous condition. So, if you think of, imagine of this, suppose you have this wild type genome, and you have a mutagenesis like tDNA mutagenesis. So, what happens that if your tDNA gets inserted, so the tDNA, it is coming from the agrobacterium, right? It is a part of triplasmid. So, tDNA is something which gets inserted into the plant genome, and then along with that, originally there are other genes like auxin cytokinin that used to get transcribed, and that leads to the formation of gall. And the one which is being used in the agrobacterium system in the biotechnology is the gene without having those. It's a disarmed vector that is used, right? Only with the DNA, tDNA portion. So anyway, tDNA, a sequence which is going to disrupt the genomic DNA, that if it is going to sit in your space, in your gene of interest, then what will it do? It will disrupt your genomic DNA, right? And if so, what we do is basically we use the primers in the sense it depends on how you are designing the primer. So if your forward and reverse primer are within the same within the gene itself, then you, and if there is no tDNA insertion, then you should be able to get the wild type band. So wild type band means just simple band, just think of it that it gets amplified and it's giving you a single band. Okay, but if you have this tDNA and if you're going to design one of the primers, suppose the reverse primer for specific to this tDNA, then what will you see? Then you will see that this if you have this tDNA, it will get amplified. If you do not have any tDNA, so and if there is and you are going to amplify with the tDNA specific primer, then what will happen? You will not be able to see any band for that. Is this clear here? Is it clear here? No. Yes, yes. Okay. So let me draw. Okay. This is your wild type gene. That is not, suppose it do not have any tDNA insertion. And this is another one which has got your tDNA. Okay. So why I'm talking about this tDNA is, suppose this tDNA insertion has led to a mutant gene which you actually want to see. The mutated one is the homozygous or heterozygous. So here we can see you have one copy of wild type, another copy, that one copy has got disrupted with the tDNA. So this is a heterozygous condition, right? So one is the intact allele, one is the mutated allele. Fine. So now, but how will you know in the system that whether it's a homozygous or a heterozygous condition? So what you can do is you can design primers. Primers in the sense, you know, the small fragment of DNA that can bind to your target gene and then lead to the amplification. Amplification is in the final signal you get, right? So if you design one primer that is only to your wild type, so you will get and band for your wild type. Fine. Now, if you design another primer that is specific to your gene along with your tDNA, so you are designing a primer between your gene and your tDNA. So what will happen if it is a heterozygous system? So you are doing actually both the primer. So what will you get? You will get the amplification for this as well as you will get the amplification for this as well, right? So in that case, this condition you will get for both the bands. I mean, for both the primers, both set of primers, you will get bands. But imagine it is only homozygous for your tDNA insertion. So what will happen? This is your tDNA. Another set is also tDNA. So in that case, you will be able to get amplification only for this set of primer. It is here, but you will not get for this set of primer because wild type is disrupted here. Okay. Now if we imagine the another condition where it is the, yeah, only wild type. So if it is only wild type, if it is only wild type, then there is no tDNA insertion, right? And if you are going to use a tDNA specific primer, then it will not be able to amplify. It will only be able to amplify the wild type one, and there won't be any band in your tDNA specific primer. Okay, is that clear now? Yes, I hope this is clear. So the markers are basically, there will be a specific, those are the specific regions against which you design your primers. Hmm. So markers are present through in your genome in specific regions. You know where it is present. So you know which sequence it is, right? So accordingly, they're talking about it, dominant or codominant. If you're able to differentiate, that is known as the codominant marker. Okay. Then, what is the question? A molecular marker co-segregates with mutant phenotype of your interest. Which of the following statement is false regarding this observation? Okay. Try to answer this question. Okay, what is this co-segregation means? You have heard of linkage, right? Okay. So linkage is like when two genes are present so close to each other that they do not I mean, if they do not follow the Mendel's law, that one, that the independent, no, the law of independent assortment, segregation, sorry, law of segregation. So what happens that we know that if two chromosomes, so if you know about Mendel, that he has worked on seven different kinds of or the seven traits, different traits, right? So which were, and he was lucky in the sense that all the genes responsible for that trait, particular trait, were present in different chromosomes, all the different chromosomes. So they all segregated independent of each other. There was no linkage in the same. So what happens that if you know about that recombination, right, during the meiosis, what happened? So two, we know that if two genes are too close to each other, then they might actually uh, what we say, they might get passed on in the same pattern, so they might not be uh, it might not show its own independence. So what happens that to break that thing, there is something known as recombination that happens, which breaks as part of your chromosome and then rejoin, so that your chromosome has the genes get the flexibility to segregate independently. Right. So as it's very question is very obvious here that you're saying that a molecular marker is co-segregating with the mutant phenotype of your interest. So definitely they are present in a very co-segregating. They went together. They're not independently segregating. So that means they're present together to each other, and as a result, they are actually segregating along with the gene. So that's why the so now you can tell which is the answer, right, from this. So, but the thing is, they're asking which is a false statement. Yeah. See, the molecular marker and the mutation present on different chromosomes. Okay, fine. Just a second. This is the week one, week two. This week two. Oh, I have already answered. Anyway, we can. It's not only about the thing is. The session is not only about the specific answer. It's mainly about the concept and discussion about it. Okay, so it's okay even if it is now what is it marked. So which of the following techniques can be used to detect the organ specific differential expression pattern of a transcript? You all know that this is northern blot, right? For okay, so here is the thing is the differential expression pattern of a transcript. The transcript means the RNA. So DNA is forming the RNA. RNA is known as your transcript here. So we know that for RNA we use northern blot, right? So you know what for western blot? One sec, just a second. Yeah, protein. Southern. Which one? Yeah, yeah. DNA. Southern. I don't know Eastern. Is there anything like Eastern? Okay, what is that Eastern blot? I also don't know about it. Okay, right. Hmm. It is okay, but yeah, maybe they're saying that Eastern blotting is a biochemical used to protein trust. I don't know about it. Right. Okay. It says that some modification, translation modification. I'm not sure. Okay. Fine. I'll just hide this answer right now. As of now. So, match the following techniques with the analysis that can be done with. Okay, so microarrays, RNA in situ hybridization chip, and translational fusion construct. So let's see what is microarray. Any idea about microarray? Okay, so microarray is something that you see the gene profile, the global gene profile, we say. So, your whole genome is producing many genes. So, if you want to know, suppose just a very simple example which is used for this microarray understanding is that suppose you have normal cell, you have cancer cell. You want to see that which of the genes are getting upregulated in the cancer cells or any other disease cell compared to your normal cells or healthy cells, if you say. So what is being done is, of course, you want to. So your target is to check what are the transcripts are getting upregulated, right? So what will you do? Okay. Anyway, I think let me see if I have the slide for that. Oh my God. No, I don't have the slide for that. I can use. Let me take one. So anyway, so what is being done is that so there are use of labeled RNAs. Okay, so in one cases it is used. Suppose for healthy cells you are using one color, and then for your cancer cells you are using another color, green. I don't know how much I can actually tell you from by just saying. So, if you have green, then if you have blue, then and in the same you do what you do is mix both the RNAs, okay, and you have a specific probe for specific probe along with your tag for the gene of, I mean, the transcript of that, and you see that which is the signal, which signal is predominating. Okay, so it is in the cancer cell or it is in the wild type cell. So by seeing the color code or the colored signal, you actually decide that which is the. So if something is suppose is more in cancer cell but it's less in wild type cell, then that the signal for the cancer cell will be high, right? And then for that color you will be able to see, and from that actually it is said that for that specific gene it is more in the cancer cell. Okay, if possible, next time I'll try to put a slide and explain in that manner. Okay. Anyway, so whatever it is, so microarrays is basically for the your gene expression analysis globally. So by now you already know that microarray is for which option is option 3, right? Okay, next is RNA in situ hybridization. Have you heard of it? I think you might have heard of it from Sriram Yadav sir a lot about it. Any answer? What happened? Any answer? Okay. So RNA in situ hybridization is something which is used for the spatial, or what is it called? The positional information about a transcript. So, okay, I'll just show you that one slide I have. Anyway, so what happens is that, suppose, okay, so the shoot apical meristem I was talking about, right? So what happens that in the shoot apical meristem itself, there will be different zones. Hmm. So suppose this is a central zone. So this is suppose the peripheral. Hmm. And then is in the is your the in the primordia, the leaf which should be forming the leaf, there are certain zones. So in different zones, there are different tissues present. Yeah, sorry, I'm tissues in the sense I mean there are different types of RNAs will be present, because you know that is we know that our genome has all the DNA that has the ability or capability to form all the RNAs. But what important is that differential expression of them, the spatial expression you can say. So the gene which might be active here, it might not be active here. Okay, because of the we know the about, if you know about the epigenetic things, that in some part some gene will be more active and some other part it might not be active because of the chromatin modification, and there could be many other factors. It's not only on the chromatin modification, it could be that certain protein might be present there, it's not able to letting it to express. Okay, so even suppose in certain cases you are producing the RNAs, but it's getting degraded by any other protein or any other, if you heard of the microRNAs. So there are various things can happen. So the dynamic of different RNAs keeps changing throughout the tissues or the throughout the plant we see. So what happens that to so whenever the problem with the RT-PCR, if we talk about, is that when we are doing the real-time PCR, what we see, we're seeing the transcript level of a specific gene, right? So what happens is that we're taking the whole tissue maybe and checking what is the level of the transcript. Okay, is that transcript getting high or low or whatever, is the fold change is significant or not, something where maybe you're comparing with some other wild type. But what we miss in that is that what is happening in this tape, what is happening in this tape, in this tape, in this regions. To know that specifically, so there is another method, that is the RNA in situ hybridization. What it does is that you have your targeted mRNA. So you have your mRNA, suppose the gene which is producing the RNA, you want to target that RNA where it is being expressed. So what you are doing, you are actually labeling or giving it another mRNA complementary to it, or RNA which is labeled. And this label, it could be through any kind of product, if you're getting, you can react, or any kind of signal it can give, and you can detect where it is present. Okay, so your transcript might be present here, and when you are giving your labeled RNAs, they are going to bind to it and they're going to give you a signal, and that way you are actually able to look it where your mRNA or the transcript of that specific gene is expressed. It is in the tip, it is in the in the apex, or it is in the base, which region. Okay, so that is the RNA in situ hybridization. Is that clear? Okay. Next is chip. Chip, all of you know, right? It's very common. What is chip? Come on, guys, interact something. Okay, chip is the chromatin immunoprecipitation assay, right? So, what you are doing here, your protein of interest, you are precipitating that with the antibody against it, and then you are checking, and then you are checking the DNA sequences to which it was binding. Okay, so you're precipitating and then fragmenting your DNAs, and then later on through if you think that a specific target, if you're searching for a specific target, then against you can use the primer against the sequence and see if there is any enrichment or not. And so that is known as chip assay. Next is the translational fusion construct. So translational here means the translational product. Translation the product means what? RNA or protein? Which one is the product? Yeah, so protein. So that means translation fusion construct means you are adding some construct or any kind of reporter. It could be reporter or any reporter could be anything. Okay, it could be signal, it could be enzyme, anything. So that is means you are fusing some reporter with your translational product or protein. Okay, now based on my explanation, could you please tell me that what could be the answer from it? Yes. First, first is it? Is it? Okay, let me check the chat. What people are answering? No answer, but yeah, it's already I have written first. So some answers could be wrong. Don't follow it blindly. Yeah. So yes, as I mentioned, that microarrays are for global gene expression analysis, and second is the RNA in situ is the spatiotemporal. Yeah, this is the word I was searching for. Spatiotemporal gene expression means according at which you can. So what happens that is, as I told you, that it's very transient RNA expression. So maybe your 5-day old seedling is showing some expression, but 6-day old seedling may not show the specific expression. So at the time point there could be also variation. So for that spatiotemporal gene expression they have mentioned. And then chip assay is the DNA protein interaction, right? And the translational fusion construct is the subcellular localization of a protein. So as you are going to fuse your protein with a specific reporter, you can actually check at which compartment of your cell this protein is being produced. Okay, so and then which of the following enzyme is commonly used to partially digest the tissue for better penetration in RNA in situ hybridization? It is a proteinase K. It's a protease which is probably get rid of the unwanted proteins to better binding of with the RNA. Okay, that is something very straightforward. So which of the following statement is false regarding a transcriptional fusion construct? Yeah, which of the following statement is false regarding a transcriptional fusion construct? Here it is the transcriptional, not the translational. So, okay, anyway, So whenever we are talking about transcriptional, there's something called promoter that comes. So as I was talking about the condensation of the chromatin, so it sometimes depends how condensed is your chromatin. So if your gene of interest is in that condensed region, it might not get expressed. Okay, so it's basically it depends on how, whether your promoter is free to access other transcriptional machinery like other proteins transcription factors, whether it can access through them or not. So if it is free or open, then it can access that, and then it can lead to the transcription of the gene, right? So yeah, so the thing is, which of the following is false regarding this? So what they're talking about is the we get to know the tissue where the promoter of the gene is getting activated. Okay, and it consists of promoter or upstream region of a gene along with the reporter gene. We get all the information about subcellular localization of the protein. So which of them is the wrong answer here? Okay, someone is answering. None. Why not none? It is actually there is an answer. See, I would like to explain it. So they are saying that regarding a transcriptional fusion construct. So if it is a transcriptional fusion construct, then we cannot get any information. Yes. Yeah. Yeah. It is. Yes. Yes. Yeah, so we cannot get any information regarding protein. That would be translational fusion construct, right? The previous one which we read about it. So yeah, so the first one with promoter activity, promoter is getting activated or not, that will definitely tell you, because promoter is the one which is the first step for, I mean, promoter has to be active, right, for the transcription. Second is the consist of promoter or upstream region of a gene along with the reporter gene. Consists of promoter, okay, upstream region of the gene along with the reporter gene. If you have a reporter, it will get to know. Fine. And definitely all this is not true, so C is correct. Next question is, which of the following techniques can be used to study protein-protein interaction? Okay, any guesses can work. But here you can already, I have already talked about the other assays. So you can actually, if you rule out, then also you can get an answer from here. Yeah, yeah, answer please. You can write your answer as well. Yes, ma'am. That's the correct answer. Okay. Yeah. So cheap assay, I just mentioned that it is about the the protein and DNA interaction, right? So protein, you were precipitating, immunoprecipitating the protein and checking for the which DNA sequences it's binding to. So it's getting ruled out. Second option is the East 1 hybrid assay. That is also not, I'm not sure exactly what is that. I think that is RNA and protein interaction, probably. I need to check this. And the third one is the RNA in situ hybridization. Just now we talked about it, that it is about the RNA interaction. So it cannot be. So the protein protein interaction is the East 2 hybrid assay. You can get the answer by ruling out, but let's see what it is. So yeast 2 hybrid assay is an in vitro assay. What happens is that if suppose you have two protein of interest, protein A and protein B, I can just quickly use my PowerPoint. Yeah. Okay. mute yourself, ma'am. Okay, what I was talking about is to hybrid assay. So suppose you have a protein A, okay, and you have another protein B. You want to see whether this protein interact with each other or not. Why that is important? Because what happens in many cases. Okay, let me mute. Stop. Ma'am, please mute yourself. Yes. Okay, guys, am I audible, right? So anyway, so suppose you want to study the interaction between two proteins A and B, and I was telling you why it is important, because what happens in many of the cases in the in vivo system, there actually a complex of protein forms, and that actually does a function. So many cases what happens, a protein A alone cannot do its function, it requires another partner to come in and join, and then some conformational changes happen, and then it gets activated and can do the function. Okay, so many cases it important to know whether two proteins are interacting or not. So what is being done here is, so how will you get to know? The thing is, we use a system called yeast system. Yeast, you already know, it's an unicellular system. So what happens is that there is another transcription factor called suppose it could be anything other also, but it is a gal4, suppose. So what happens is a transcription factor. So transcription factor, we know that it is a factor or a protein which can actually start the transcription of a specific gene. Okay, so what they have done very cleverly is that they have cleaved this gal4 into two parts. So basically what happens, the transcription factors will have two parts. One is the activation domain, another is the binding domain. So they have made it two part. One of them will be containing the activation domain, and another part of it will be having the binding domain. Okay, so binding domain, what it does, it binds to the DNA, and activation domain, it activates that gene or the transcription of the gene. Okay, so what now, if you want to study the interaction between A and B, you can actually tag your protein A with this, and you can tag your protein B with this. How you will be doing this? Because you will already having vector which will be having this activation domain sequence, and there will be some cloning sites where you can clone your gene of interest, suppose A, and in another vector you can clone your sequence B, where you will be having your binding domain. Okay, now the idea is that this two a part of the gal4 cannot actually interact to each other unless it is brought together. So how will that happen? Suppose this your A and B are true interactors, then they will interact and come close to each other. And when they will come close to each other, what happened later, that these two proteins will also come close to each other, and then they form a complete set of it. And because of it, it is going to form a complete set. It can actually lead to the transcription of those genes, and those genes are actually the reporter gene, which will tell you whether this interaction is happening or not. If this interaction is happening, your reporter gene will get expressed, and that will actually tell you that these two proteins are indeed interacting. Okay, is this fine, clear? Guys, am I audible? Yes. Okay. So the thing is, it's 7 o'clock, right? So there is 7 to 9. There is one more session that will be taken, I guess, by Partha. So you guys can attend that class. So I have to actually log off now. Okay. So see you in next week, probably with some more assignments. If you have doubt, you can. I mean, you can, if you have doubt, you can ask later then, and of course, you can follow the next class. Okay. See you next week. Thank you. Thank you, everyone, for joining. Yes. Please join the next class, 7 to 9. If you that much other questions will be answered there, and if you have many other doubts, that also.